Welcome to part 3 of putting it all together, final edits and visual polishing. As an overview, we're going to look at labeling charts and tables properly, using captions, numbering, and formatting standards, and polishing your presentation. So what is the point of polishing and making pretty graphs and figures? Well, it's that while our writing is the primary presentation of the data and should be able to stand alone, the human brain loves variety and visualization. So for that reason, having all of your writing and findings and graphs and tables without proper and thoughtful organization can make the work really unapproachable. By providing organization, structure, and guidance to the reader on how to naturally follow the data and how you present the data is very important. A well-captioned and organized figure is an invaluable tool in the pocket of any data analyst and anyone writing a paper, because this is how you will make your results approachable. And in many cases, if our results are not approachable, the audience is a lot less interested in what we're saying, and we want the audience to be interested in what we have to say. Choosing your figure components is an extremely important aspect of polishing the final paper, because it requires you to understand what it is you're trying to communicate so that you can choose the best method to do so. So if you're trying to explain a process or a method, things like a flowchart, diagram, infographic, illustration, timeline, those things are going to be the most helpful. If you're trying to compare and contrast or show change, our bar, box, and line plots are classics. Stacked graphs, pie charts, histograms, those are going to be your bread and butter. If you're looking to establish a relationship, something like a network diagram, a heat map, Venn diagram, mosaic chart maps, etc., there are way more types of plots than I could possibly mention here. But those are going to be the ones that you'll use to make connections for your audience. If you've already selected a journal to submit to, please refer to their submission guidelines for specifics on the requirements. This will include font requirements, text size, number of figures allowed, size of figures, how to write and arrange captions, etc. Let's walk through an example of looking at submission guidelines. We're going to look at a journal called +1. So what I would do is I would Google +1 submission guidelines. When I follow the link, you'll see something like this. They have a section for style and format, manuscript organization, parts of a submission, pretty much anything you could possibly want to know about the proper way to format for submission to this paper. I'm going to go to parts of a submission, and I'm going to find their figures and table section. Here they say, for example, do not include any figures in the main manuscript file. So what a lot of times they would like for you to do is to have your written manuscript in one file and in another file, to have your figures in the order they would appear, all properly formatted, just separate. They also tell you that they would like you to cite figures in ascending numeric order at first appearance in the manuscript file, which is important. Then they go into writing figure captions and how they want those referenced. They also have here guidelines for figures and requirements reporting blot and gel results. So if I follow that, they give you a figure preparation checklist with guidelines, figure file requirements, formatting them, and they're giving you resources to make sure that your figures are converted into acceptable formats. So while this is a bit of a administrative end goal part of the project, it is important to know where to go to get this kind of information. So let's talk about some figure basics. Figures should be neat, aligned, all same font, all same text size. You want these to be presentable and attractive and professional looking. So by aligned, what I mean is that all the edges of your panels, which I'll describe in just a moment, should be along the same lines. You should use as much real estate as possible, and we'll take a closer look at that later. So every individual asset within your figure is referred to as a panel. So every graph is a panel of the figure. That being said, you should have each panel labeled A, B, C, D in order. And it's going to be in the order that they do appear in the text. And you'll want to use the figure to represent only your most relevant findings. You also want the figures to be numbered and presented in the order of appearance in the paper. So for example, if this here is our figure with panels A, B, C, and D, they might be discussed like gene expression as seen in figure 1A shows treatment during the drug, blah, 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 patient demographics, figure 1B shows blah, 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 responses to the drug, figure 1C. And you'll want to have informative but concise captions. So for example, often you might have a figure caption that starts with sort of a title like figure 1, treatment with drug increases protein X production, leading to positive responses from patients. And it should include things like your number, like the N involved in your data, and the statistical significance, how it was determined, and what the various p-values are. So let's talk about plots. This is going to be plots are going to be one of your most common panels in a figure. So let's talk about how we should be formatting them and some little notes on how to make them look their best. So this is a figure from a paper by Rafat et al., published in Cancer Research in 2018. I like this figure because it actually uses a number of different types of media or asset types to communicate their data. So first we get this schematic in figure whatever it is A. And then we also have a photo of some organs and how they looked under a luminescence reading. And then we have our various plots. So let's talk more about that. So first of all, I would strongly recommend removing grid lines. So grid lines are those little lines that go across your plot like this. They end up looking kind of awkward and often just don't have the most polished look. Next, axes titles. This is one of the most important things, and it will get you dinged immediately if you do not have them. Axes titles should describe what exactly is being measured here. So in this paper, what was being looked at was cancer cells that had a luminescent tag migrating to this site over here. So while the luminescence is a measure of the migrating cancer cells, it would be improper on the axis to put cancer cells. So, because that's not actually what's being measured. What's being measured is the luminescence from the cells. And it's very important the unit is included. So the unit here was photons per second, based on the instrument that was used for the reading. So being descriptive, but not too descriptive, with your axes labels is very important. Also down here, making use of formatting to make this as neat as possible. So instead of having control MFP, control peritoneum, control muscle, which would have been a lot of space, the authors have the same, just simple indications and an overall definition. On that note, color is one of the most underrated formatting tools for plots. For example, here, you should always have a definition of what your colors mean. But let's imagine for a second there wasn't one. You would still probably look at this and say, oh, the black one is probably some sort of control and the red one is some sort of treated. Color is very, very powerful. It's very attractive and it makes your plots interesting to look at. Especially if you have more than one condition, I would say a color is a must. Another thing is that if you can carry the same color theme throughout your figures, it creates such a polished feel for your paper and your figures, and I think it typically goes very well. Next, you have to include error bars. So error bars are going to represent, such as this will represent like our mean and then the error. Without these error bars, it can be a little bit difficult to tell sometimes where exactly the mean of the data is, and that can make it difficult as a scientific audience member to interpret that data. So in this case, you can see we have our means here, and you can tell, oh, well, there's a little bit of a difference here, but overall, these are very similar. However, if we come over here to the irradiated, we can tell pretty much immediately upon looking, oh, the mean is so far up compared to the mean of the control. And the error bars, so essentially the stretch of error based on our data still doesn't overlap with the error or the mean of the control data. So that can let the audience look at it and just on a quick glance say, oh, yes, this data, I believe this data, I believe what I'm seeing here. Next, significance. So significance is going to represent the results of statistical testing done on your data. And this is how it's often represented with these stars here, because it's very clunky to have the actual p-value, although it's never done. You just have to make sure that these stars are defined in the figure caption. So, if 1 star is p equals 0.05, or 2 stars equals p less than 0.001, that just has to be stated in the caption. So here we're going to look at the order in which panel labels are presented. So overall in the paper, this is figure 2, and it is the second figure that is referenced in the text. So in addition to figures being appearing in reference order, so should the panels. So figure 2A is the first panel described, and this is the mouse talking about the memory fat pad, the schematic of the experiment, B, C, D, E, and F, and so on. So you have to make sure not only that you include labels here in the panels so that they're identifiable in the text, but also all of your panels should be referenced in the text as well. Now let's talk about captions. In the captions, of course, you're going to define what the figure you're looking at is. It's going to have probably like a little figure title, but we should also talk about things included like error bars and significance. So if you look at this figure here, this is panel E from figure 2. We're looking at control and irradiated tissue samples and the luminescence from the migrating cells. In this figure, we obviously have error bars, we have p-values. So as I stated, those should be mentioned in the figure caption. If you look into the figure, let's go down to E and this caption here, you can see that it states how statistical significance was determined. It was determined with an ANOVA, and the p-values that coordinate with the stars that were used. It also tells us the error bars show 95% confidence interval, and that it says in S it was non-significant. So the final arrangements of your figure are going to be a little pedantic. They might seem that way at least. But really what you want to do is to have the most polished looking figure that's going to attract attention and is going to present your data in the most authentic and clear way possible. So let's go over how I address my figures when I'm editing them. So first I'd like to look at the space that's being used. A lot of times this might include if the text is a bit small, such as in figure B, panel B here, or if things don't fill the space that they could, such as panel A and panel D, we want to make sure that we don't overcrowd the figure, but that we do use the space available. So for example, here in panel D, we can see this is quite crowded, and I have a ton of unused space next to it. The text is quite small in panels B and C, and looking at C, this text that says merge, it's a bit extraneous. I think looking at it, we all understand, or at least in the field, they would understand what that is. In addition to that, panel C, the images are in no real particular order. So I think it would make the most sense for the figures to go from the least signal to the most signal, since we have that sort of inherent order here in the figure, but that is not how it's been ordered. So it's a little bit hard to follow the data. So for panel A, you can see here how it's been changed a bit to make things a little bit more compact, and it takes up more space here next to panel B. We can see here in panel B that the text was made simpler and larger. The color was removed from the text, and that might be due to a request from the formatting for the paper, for the journal that they submitted to. So I'm not sure why that was an edit that they made, but the text is certainly larger, which is better. Here in panel C, you can see now how going from OLIG2 to FOXP2, we see an increase in signal, and we see that in order. So comparing the control and the miRNA across these various stains is a little bit more aesthetically pleasing. And finally, in panel D, we did go ahead and stretch out that plot, which made the plot less crowded and took up that unused space. So there are lots of places where you can make the panels, the components of your figures. Plots, you will likely make these in your spreadsheet program and then copy them over. There's also the opportunity to maybe edit them aesthetically in things like Adobe Illustrator or Inkscape. We'll talk a bit about that. For schematics, you might find the icons and art or pieces free through sources like the NIH BioArt, MIT Icon, SciDraw, the Noun Project. For the sciences, there is also BioRender, although BioRender unpaid is actually quite limited, so I prefer using one of these. Flowcharts you might produce in a spreadsheet program, but also PowerPoint, Canva, and other programs do this very well and very aesthetically. For images, you should make sure that they are your own and that they have minimal touch-ups and manipulations. Again, a lot of journals have rules about how much you can do to an image for submission. The key is that you just don't want to be altering the information that's there to prove your point. But the journal will let you know if you're allowed to do brightness and contrast, if you're allowed to crop, those sorts of things. So you can put these panels together. The assembly can be done pretty much wherever. You can easily gather assets, move them, align them, and resize them without introducing noise and distortion, so without making it a quality looking image. You could use PowerPoint, which is easy to work with and widely available. It also has tools for alignment, and you can save and come back to it. Similarly, you can use Google Slides as well as a free option. You can also use a vector editor like Inkscape. Inkscape is free. The nice thing about Inkscape is that you get to work with layers, which means that you can have your text on one layer and your images on another. It also has a variety of alignment tools to help align your panels and make sure that your figure looks nice and tidy. And you can save and come back to it. And there's also a lot of export options. And because it is free, I highly encourage it. It's a lot like Adobe Illustrator, but without having to pay. Finally, congratulations. At this point, you are well on your way to completing your write-up, and I think that you should be very proud of yourself. Make sure to take your time, make sure to have a critical eye, and good